like Stein said, only 6 months ago I gave a lecture here, and some of you might have been present and so on. And you know, 6 months in science is not a very long time. So I was starting to think, hey, how can I be original? How can I show new results? Thanks God, just before leaving to China, we submitted my lab submitted the paper to a journal. I will not say which one. Maybe it brings good luck. And I will tell you today about this story, which is very new, unpublished, and maybe you'll never see it published, but at least you'll listen to me. The general topic of my lecture indeed will be stem cells. And whenever we talk about stem cells, obviously most of you, especially students, possibly will think about stem cells in embryo life, in embryonic development, because certainly stem cells are extremely important in embryonic development. However, I would say they are equally important in the adult organism, especially in organs and tissues that have a very high self-renewal, that are always renewing every day and so on. Think of our blood, made by bone marrow cells. Think of our intestinal cells, and we'll talk a lot about that. Or our skin, our largest and biggest organ and so on. These are all tissues where every day we lose millions and millions of cells. Obviously, these cells that we lose should be replenished, should be substituted by new cells. And this is what adult stem cells do. The problem is that the characteristic of stem cells are also the characteristic that cancer cells like very much. And that's why we now know that cancer stem cells are really the beating heart of a tumor. And that is really something that we should target to cure cancer in our patients. And this is the paradigm of the beauty and the beast. The beauty in our normal, healthy body, but the beast in cancer. And obviously, every tissue in our body is renewed at different rates and so on. The rate at which the skin will renew will be very different from the rate at which the gut will renew. In the skin, we know that there is a structure called the bulge, where stem cells are resident, and from here they go here to form the hair, but they also go to form sebaceous glands and also epidermal stem cells, so the stem cells that make the skin. But in general, all stem cell niches have the same characteristic. They have a hierarchical structures, so this is not a democracy. Not all cells are equal. Some cells are more equal than others, if you want. And stem cells are on the top of this hierarchy. They can self-renew, and while they progress, they will still commit, and then eventually terminally differentiate. So the two characteristics of stem cells are certainly their capacity of self-renewing and the capacity of differentiating. And by self-renewing, we mean that every time the stem cell divides, it makes sure that at least one of the two daughter cells is a stem cell, right? And this works in a very simple way. In fact, we know that stem cells can divide in an asymmetric way or symmetric way. Symmetric when they generate two identical daughter cells, and asymmetric when they generate one daughter cell that is differentiated and one daughter cell that is stem cell. And this is an example of a muscle. You see, if the mitotic axis is parallel to the lamina propria, then you have a symmetric division, because both cells will still be in contact with the lamina propria, while if the division is perpendicular to the lamina propria, then one cell will remain a stem cell, and one cell will commit, will differentiate. And the amazing thing to me is that each of these stem cell niches is regulated by specific signaling pathways, like the wind, like sonic hedgehog, and like notch. And the interesting thing is that every time one of these pathways is mutated, you get cancer in the specific tissue type. For example, wind signaling is characteristic of the colon stem cell, and it's also the pathway that, when mutated, can give colon cancer, and so on and so on. Now, we'll talk a lot about the intestinal system. So let me talk to you a little bit about that. As you know, our gut is covered by a monolayer of epithelial cells, and this monolayer forms crypts, which are invagination, and this structure, dome-like structure, which are called villi. And at the bottom of the crypt, there are some stem cells that divide, and one of the two daughter cells will move upwards, form transient amplifying cells, which are cells that are rapidly proliferating, and then eventually they will move upwards and differentiate in the 4 or 5 differentiated type of the gut, and then eventually become apoptotic at the top of the villus. The entire cycle takes 5 days. So within 5 days, our entire epithelial lining of our entire gastrointestinal tract is replenished, which, if you think about it, is quite spectacular. In fact, it has been calculated that we lose every week about half a kilo of apoptotic cells. And with such a high dynamic system, you understand that the one important factor is to reach an equilibrium. Because every time a new cell is made here, a cell should die here. Pretty simple, right? And what keeps, indeed, the equilibrium is the wind pathway. So the cat here is wind that indeed can keep this equilibrium between cell division, cell proliferation, and cell death and apoptosis. In fact, it's much more complex than that, but we'll go into that in a minute. Let me now show you in a video how this actually works. So this is the gut. Now we are going to blow in. This video was made by Pixar, the same company that made Toy Story, for those of you who knows Walt Disney animation and stuff. So these are villi, and around each villus, there will be 6 crypts. Now we are going to dive into the crypt. There we go. And here are the stem cells that are called LGR5 plus cells because they express this marker. And if you label these cells in blue, with beta-galactosidase and so on, then you leave the mouse go. This is the mouse experiment, obviously. And then you will have that all the descendants from the original blue cell will form a ribbon, meaning that indeed from this cell, all the differentiated cells of the gut are derived. And as I told you, they start differentiating into enterocytes, goblet cells, and so on. Move upwards, reach the top of the villus. It's a bit slow. And then when they reach the top of the villus, they will commit suicide. They will go into apoptosis. And again, the entire cycle takes about 4 to 5 days. So pretty spectacular high level of dynamic and self-renewal. Okay, and as I told you, what keeps this very dynamic system is the wind signal transduction system. And it's really a very simple signal transduction pathway. It's a on or off system. So when the signal is absent, the signal being the glycoprotein wind, then a complex is formed intracellularly between beta-catenin, APC, and other proteins. And the important part of this complex is that beta-catenin will be phosphorylated at serine threonine residues, and this will trigger the degradation of beta-catenin. So without signal, there will be degradation of beta-catenin. But when the signal is present, the wind glycoprotein, it will bind to its natural receptor and, in a way, prevent that this destruction complex is formed. So now beta-catenin is happy to accumulate in the cytoplasm and eventually move to the nucleus, where it will complex with the TCF transcription factors and activate a bunch of target genes. So it's an on-off situation. No signal, beta-catenin is degraded. If the signal is present, beta-catenin goes to the nucleus and works as a transcription factor for a number of genes. And you will not be surprised to hear now that the wind pathway is very active at the bottom of the crypt, and it's not active as you go up there. So there's probably a gradient of this ligand being present very at high concentration here, but as you move upwards, there will be less and less wind proteins, so less and less wind pathway activation. Very simple system, if you will. And this helps you also to understand cancer because, okay, again, when the signal, when the street light is red, then a genetic program is activated in the cell. That is a genetic program of differentiation, of migration, moving upwards, and eventually of suicide, of apoptosis and so on. But when the signal is present and the stop light is green, a completely different genetic program is activated. This is a genetic program of stemness, of proliferation, of self-renewal. And these two, again, must be kept in an equilibrium. And you can understand if there is a mutation in one of the genes that make up the pathway, then the cell might be stuck in the on status. So the cell doesn't make any more difference whether the signal is present or absent. Now it's always signaling. The street light is always green. So this program, genetic program of stemness or proliferation, cell renewal, is always activated. And this is everything you need to disturb this original homeostatic equilibrium. So instead of having everything in perfect equilibrium, you will have more cell division and less apoptosis. And this is all you need to get formation of a cancer. And this is very important, especially for the students, because even textbook writes that cancer is a disease of proliferation. It's not. In fact, the rate at which cancer cells proliferate is very similar to the rate at which our normal cells, our normal stem cells proliferate. Cancer is a disease that results from disturbance of a homeostatic equilibrium. You don't need to activate proliferation to get a cancer. For example, in this system, if you make the cell resistant to apoptosis, and voila, this is just the same thing. Because being resistant to apoptosis, the cell will stay in the system, will not be exfoliated and so on. The same thing for example, migrating upwards at slower rates. Again, if the cells migrate at a slower rate, the results will be the same. So there are multiple ways to go to Rome, and not only proliferation as the only solution. Okay, let's now zoom in into the real crypt, because the two essential elements of the stem cell niche in the gut are the stem cell, that we call an LGR5 plus stem cells, because this is the gene that characterizes the cells. And also very important is the niche cell next to it, which is the Paneth cells. The Paneth cells makes the life of the stem cells possible. So these two must be together, otherwise the system doesn't work. Because Paneth cells provides the stem cells with a lot of negative and positive signal to indeed maintain this homeostatic equilibrium. And in the lab, we have developed a number of tools to, in vivo, to be able to follow these cells. So we have all kind of Cre-Lox mice, where we can label differentially the green cells here are LGR5 cells, and the red cells here are Paneth cells. So in vivo, in the mouse, we can really follow these cells entirely in vivo, which is a very handy system. Now, I have to open up a small parenthesis about Paneth cells, because they are the cells I am in love with. I really love these cells, and every day we work, we find more fascinating aspects of these cells. But first of all, they were originally identified by this pathologist, a German pathologist called Paneth, and indeed, because they were containing a lot of granules of secretory molecules that control the bacterial flora in our gut. They are key mediators of host-microbe interaction. In fact, they are very important in inflammatory bowel disease like Crohn's disease or ulcerative colitis. During development, in fact, during the very first week of life, when we start drinking milk from our mother, they make sure that there is crypt formation. When we are born, we don't have crypt. We have a flat intestinal epithelium, but because of the formation of Paneth cells, and in contact with the very first milk of the mother, we form de novo these crypts. And this is because Paneth cells. As I told you, they are the niche cells. They provide niche support to the stem cells. And this will be the theme of today's presentation. They, in fact, behave as quiescent stem cells. And we'll get to that in a minute. In the colon, so Paneth cells are typical of the small intestine, but there are Paneth-like cells also in the colon. And last but not least, last year we published in Nature this paper showing that if you do metabolomic analysis of Paneth cells and LGR5 stem cells, while the Paneth cells have a very strong glycolytic, so Warburg type of metabolism, the stem cells are in fact very much mitochondrial, so they metabolize, they get their energy through the mitochondria and so on. So, fascinating, multifunctional, multitasking type of cell. Now, I have to show you a little bit of other instruments, tools that we have in the lab to study these cells. First of all, we use a lot of organoid system, and I'm sure most of you will be familiar with this system. Basically, we isolate from the mouse small intestinal or colonic crypts. We put them in a very special type of culture with matrigel, with no serum at all, and with some growth factor, and they form these beautiful structures with crypts and villus that are indeed the analogous of our gut. And you see how they look under the microscope. They are really beautiful way of culture, very expensive because of the reagent that you need, that they cost really a lot of money. And on top of that, you can, so this was done from bull crypts, so this does not allow you to study single cells, but you can also do it from single cells. You can isolate Paneth cells and stem cells from different mice, put them together in a tube, and they will form organoids. And obviously, if you use now genetically manipulated or modified mice, you can really study specific gene function in specific cell types by applying this system. And this system was developed by a very brilliant PhD student in my lab, Matthias Schrewer, and this is the paper in Cell Stem Cell where it has been published. Good. Now I have given you a very long introduction and some background on the tools that we need. And let's see what indeed is happening here. Because in general, we know that every stem cell niche is characterized by two types of stem cells. You have rapidly cycling stem cells, and this is what I showed you the LGR5 cells are, but you also have slow cycling cells, dormant cells. I call them always the sleeping beauty and so on. And this is a feature that I really love. This morning I learned the word. Benji, help me. Yeah, that's it. Never learn Chinese. And these are really the sleeping beauties of our body because they are always sleeping, they are doing nothing at all and so on, and only when they are asked for, only when there is an injury, for example, and they wake up, like the sleeping beauty when it's kissed by the prince, and they can become active stem cells and repair the damage and go back to their sleep. And the funny thing is that in every tissue, the skin, the mammary gland, the hematopoietic system, we have already characterized which is the rapidly cycling and which is the slowly cycling stem cell. But in the gut, this has been a very long-standing issue, and there is a lot of competitive laboratories trying to understand which one is the sleeping beauty of the gut. And let me remind you of something, because this is about our regenerative capacity when we are exposed to an injury and so on. And I don't know if you, when you were kids, used to do this, but when I was a child, I used to capture lizards, chop their tail, keep them in a box, and then, after a couple of months, like magic, the tail grows back. And you don't have to think the tails are a simple organ. No, they are extremely complex. They are made of cartilage, muscle, epithelial cells, and so on. And if you think about it, this is fantastic. And only fish and amphibians kept this regenerative capacity. It's like, imagine that someone chop up your arm, and after a couple of months, your arm grows back. Wouldn't that be that wonderful? Well, it's not like that, unfortunately. And somehow, during evolution, we have lost this fantastic regenerative capacity. Still, we have some regeneration. Think of burns, burn injury and so on. These are pretty badly injured hands and so on. And we can use stem cells of the skin to repair this kind of damage. But in my specific case, in the case of the gut and so on, the most known type of injury, of tissue injury, is inflammation. And we know that there are inflammatory bowel diseases like Crohn's disease or ulcerative colitis. Crohn's disease affecting more the ileum, so the upper GI, while ulcerative colitis is more distal. And we know that these patients have an increased risk for colon cancer. Now you should think of regeneration, the stem cells reacting to the injury by proliferating, But the price to pay for that kind of regeneration or regenerative response will be an increased risk of cancer, because indeed you are making stem cells proliferate even more. What is also fascinating about these diseases, especially in ulcerative colitis, this is something that only pathologists know, but in ulcerative colitis patient, we have a phenomenon that is called Paneth cells metaplasia. And it means that this, in the colon of these people, we find very typical Paneth cells, which is really strange, because as I told you, Paneth cells are characteristic of the small intestine. You don't see Paneth cells in the colon. And now in this patient, in a response to the inflammatory insult, injury, this patient forms Paneth cells. This is what pathologists call metaplasia, so meaning that a cell type that does not belong here appears in this organ. Good. How do we model inflammation in the mouse? Because I work with mice and so on. I always try to do as much as possible in vivo work, because I have my doubts about the significance and validity of the in vitro work, but we'll get to that in a second. Very easy model. I just add this molecule that is called DSS, or sodium dextrosulfate, in the drinking water of the mice. Easy experiment. Just put 3% in the drinking water, expose them for 1 week to that, and after 1 week, they will have this very acute inflammation throughout the entire gastrointestinal tract. The best and the easiest model that you can even think about. So after 1 week, we know exactly that within 3 or 4 days, the gut will regenerate, and we can take out the guts from these mice and analyze them, or follow the different LGR5 and Paneth cells during the regenerative response. And this is what we have done. And the very first shocking result was that upon inflammation, so when we give DSS to these mice, here are in a FACS plot, in green the LGR5 stem cells, and in red the Paneth cells. And you see that between control and DSS treated, the number of Paneth cells stays more or less the same, while LGR5 dropped down dramatically. This is the quantitative part of it. You see LGR5 goes way down, while Paneth cells stay more or less in the same numbers. That's quite shocking, but let's now see by lead. So, oh sorry, I forgot this. This is the image control. The green cells are the stem cells, and in the DSS inflamed mice, no LGR5 whatsoever. Now let's see whether the LGR5 cells still have the capacity of act as stem cells. So let's do this lineage tracing experiment. We label the cells and let the mice go for a week. So we expect these ribbons of green cells or red cells, as I showed you before in the video. We do the DSS experiment, and again, while in the control everything seems to be normal, LGR5 cells are capable of forming the entire epithelial lining, in the DSS animals, this is simply not happening. So that's really strange, because again, these mice are regenerating. They are forming a normal gut after the inflammation, but still, the most important stem cells of the gut are gone. And this is just a control experiment I will make. So, inflammation dramatically decreases the number of LGR5 plus stem cells without affecting the regeneration of the gut. So how does this work? Well, again, we use organoids to try to understand, because obviously you expect that when we are exposed to inflammation, we increase our stemness potential to contribute to the regenerative response. So we used Hultkriegs organoids to do the control experiment, so mice on normal water, and you get a number of organoids, like 50 about here. But when we do the DSS experiments, and we repeat the organoid the same, look at this. The number of organoids is increased, meaning that we are improving our stemness, our capacity to react to the tissue injury. So this is quite shocking, because we have an increase of stemness, but we lose the stem cells, the LGR5 stem cells and so on. So inflammation on its own improves our stemness, our intestinal stem cells, but at the same time it kills the most important rapidly dividing stem cells that we know of, the LGR5 stem cells. Okay? Now, this was done with organoids, let's say, so we're using the whole crypts. That really doesn't allow us to distinguish which cell is doing what. So we used other systems to understand whether Panette cells were indeed responsible for this. Well, first of all, what we noticed is that upon DSS, Panette cells becomes proliferative. They are Ki67 positive. This Ki67, as you know, is the marker for proliferative cells. And now we are looking at cells that are terminally differentiated. Panette cells are considered, if you take a histology book, they are considered post-mitotic and terminally differentiated. So they are bound to die, if you want. But now, upon inflammation, they are acquiring proliferative capacity. This is to show you how many crypts with Ki67 positive Panette cells we see upon inflammation and in the contact. And this you can also see in Crohn's patient, also in Crohn's disease, we see Panette cells acquiring proliferative capacity. Then we did this reconstitution assay, as I told you. So we took Panette cells from the DSS-treated mice and stem cells from normal mice, and indeed we found out that Panette cells really favor the entire organoid formation. In fact, it's quite extreme, because when we use only Panette cells from the DSS mice, these Panette cells that are proliferated, in fact we can get organoids. So now we can get organoids without the LGR5 stem cells. We can make organoids from terminally differentiated post-mitotic cells. This is the film of the, so we labeled these Panette cells in yellow in this case, and you see that the resulting organoids is completely yellow, which is the smoking gun type of evidence to tell you that from Panette cells, indeed, you can derive organoids. So Panette cells, in a way, become stem cells during inflammation. I told you that you have to take any kind of ex vivo or in vitro experiment with a grain of salt, because eventually what really counts is what's happening in the living organism. So what we did was to repeat the experiment in the mouse, give the mouse DSS, and follow the behavior of Panette cells. Now Panette cells are labeled in yellow. And you see 3 days, 7 days, 10 days after DSS inflammation, and you see that indeed from Panette cells we get a ribbon covering the entire intestinal epithelium, proving indeed that Panette cells can act as stem cells when upon inflammation. And this is just to show you that from Panette cells we get again LGR5 stem cells, and from Panette cells we can get all the 5 differentiated types of the intestinal epithelium. We did also some RNA-seq analysis, and indeed, when we FACS sort Panette cells from these animals, you see that Panette-specific genes, like lysozyme, defensin, and so on, are going down. So from red, they are becoming blue. Stem cell genes are going up. Indeed, they are acquiring stem cell activity, and cell cycle proliferation and so on are also going up. So the RNA-seq analysis confirmed that these cells de-differentiate, lose their Panette cells identity, acquire stem cell identity, and start indeed to behave as stem cells. I don't know about you, I think that this was pretty spectacular, because it's the first time that someone proves that a fully differentiated intestinal cell can indeed provide a stem cell identity. Now, obviously, you never end asking questions and so on, and our question was, yeah, okay, but what is the signal during inflammation that induces Panette cells differentiation? And looking at the literature, we noticed that during inflammation, a lot of this protein here, SCF, is made. So SCF is a stem cell factor, and it's a ligand for a receptor that is called c-KIT. And guess what? c-KIT is specifically expressed in Panette cells. So during inflammation, both in the mouse and in the patient with Crohn's disease and so on, there is a lot of expression from the stroma of this stem cell factor. The stem cell factor is the ligand of c-KIT, and c-KIT is a receptor that is only expressed in Panette cells. Now, you start thinking, and indeed you start reading the literature about stem cell factor. There's a lot of literature about this. It's a very important factor in hematopoiesis, in a number of systems, but it wasn't really discovered yet in the intestinal system. And indeed, by using only SCF, so now we are using only normal mice, and we are introducing recombinant SCF in the medium, we can indeed make Paneth cells form organoids, only with SCF. Proving that indeed, yeah, these are all, I'm not going to explain here all the boring, detailed experiments, you will have just to believe me, but indeed, again, by exposing Paneth cells, sorted Paneth cells, to SCF, we can again make them form organoids, only with addition of this single factor. Next question, which are the downstream pathways, downstream of SCF and c-KIT that characterize this dedifferentiation reaction and so on? We did a lot of work on this, and eventually we found out that activation of the PA3 kinase AKT pathway was really important, because that is activated by whenever c-KIT binds to SCF, and on its own, PA3 kinase AKT activates Wnt signaling. Now you can collect everything, because Wnt signaling was indeed the pathway that I told you was so important, stemness. And in fact, we can take again Paneth cells, add a molecule, it's called chiron, and it's an inhibitor of GSK3B kinase that activate Wnt signaling. And again, we can make Paneth cells form organoids only with the use of a Wnt ligand, if you will. So to conclude this part, I don't know, how am I going to do with time? Okay? We'll still need 10 minutes, I guess. To conclude this part, what I showed you was that upon inflammation, first of all, we lose our rapidly cycling stem cell, the LGF5 stems. Still, stemness is increased, and this phenomenon, is indeed triggered, or anyway helped, by Paneth cells dedifferentiation. Paneth cells dedifferentiate and again form stem cells that will trigger this SCF-c-KIT interaction, PA3 kinase activation, GSK3 beta inhibition, and beta-catenin that eventually move to the nucleus and activates Wnt signaling. This is how it works. So, to put it in writing, terminally differentiated and post-mitotic, very important, Paneth cells are capable to respond to inflammation by dedifferentiation, re-entering the cell cycle, and acquiring stem cell capacity. The rapidly and so important LGF5 stem cells are completely ablated, they are lost during inflammation. And indeed, activation of the SCF-c-KIT signaling pathway underlies the Paneth cells dedifferentiation. These are the detailed conclusions, but to me, what is really important is that this shows that there is much more plasticity in our body than we thought of. And that even differentiated cells can behave as stem cells when stimulated by tissue injury. I showed you this graph in the beginning of my talk, and if you remember, all the arrows were going one way, and this was indeed what the old dogma was. The dogma was that differentiation is a way of no return, it's a one way, that you cannot go back. Well, now we are showing that this is actually, in fact, is not true, that even terminally differentiated cells can go back to the original stem cell state. And I hope that I have convinced you, at least of the case of Paneth cells. Okay, this is important. Why is it important? Well, it is important because it means that Paneth cells may be the cell of origin of cancer in specific conditions. This is another video that I want to show you. And this is an experiment done by Hans Clevers in Utrecht, a very famous Dutch scientist. And he showed that only when introduced a mutation in the APC pathway in the LGR5 cells with a Krylov system, again, now we are introducing APC mutation in the LGR5 stem cells, only in this case, you will get a tumor. So LGR5 cells are the cell of origin of cancer in probably the majority of the cases and so on. But now I'm showing you that upon inflammation, Paneth cells can become stem cells, and LGR5 cells are eliminated from the system and so on. So it raises the issue that possibly in inflammatory bowel syndromes, not the LGR5 cells, but the Paneth cells are the cell of origin of cancer. And we have now, in fact, we are looking for a postdoc to do these experiments in our laboratory for the coming 3 years. So if you're interested to apply for this position, that will be a very exciting project where we introduce specific mutation in Paneth cells, give DSS to the mice, and see whether these mice can form tumors from Paneth cells. Okay, I'll move to the very last bit of my presentation, and it's about cancer therapy. This is the Faculty of Medicine. Eventually, we want to help the patient. We want to cure the patient. And if stem cells are really the bad guys of the cancer, well, that should really help a lot. Because nowadays, in what oncologists do, they use chemotherapy. Chemotherapy will shrink the tumor because it will kill most of the tumor cells, but if you leave behind one stem cell, the tumor will relapse, will go back and grow back in the patient, and very often in the process, cancer stem cells become resistant to chemotherapy. But if we now, especially pharmacologists, can specifically target stem cells, well, this will be the way we can cure cancer. And people have been compared this with a driving car with a tank full of gas. Now, if I take all the gas out of the car while the car is driving, the car will move for some 100 meters, but then eventually will stop. And this is what people think of, how people think stem cell therapy in cancer will work. Indeed, you are depriving the most important cells to the tumor. Is this realistic? Well, unfortunately, there are a lot of problems in this. And the main problem is that stem cells, both normal and cancer stem cells, are resistant to everything. They have developed throughout evolution a number of systems to protect their own genome, because they are the most important cells in our body. So they have developed, for example, intracellular pumps, detoxifying enzyme, and very efficient DNA repair. They are resistant to DNA damage-induced apoptosis. They have very high level of reactive oxygen species, and often they are quiescent. And you know that chemotherapy only works on rapidly proliferating cells. So if you have quiescent stem cells in your tumor, well, they will not be sensitive to chemotherapy. So how do we go about this? You know, the big pharma is working a lot in generating new molecules that are specific for stem cells. I am a little bit skeptical about that also because those drugs will cost billions of dollars, and big pharma will make billions of incomes, and the patient will only have to pay more and more money to be cured. I am more interested indeed to alternative and possibly also a little bit traditional Chinese medicine type of intervention. And I was particularly interested to the resistance of stem cells to DNA damage-induced apoptosis in relation to a molecule that is the oldest possible drug in the world, aspirin. I think I have, yes. Is this correct? Is this the Chinese word for aspirin? Yes. Why is this amazing? Well, first of all, we know a lot about aspirin because indeed it has been the oldest drug ever. Aspirin was introduced by Bayer in 1836, a long time ago. Before that, people used to make an extract of the tree cortex, usually the willow tree and so on, to fight their fever, their pain, and so on. And then in 1836, Bayer isolated the chemical compound from the tree and indeed made aspirin. And indeed, it's an amazing story. This is the willow tree. In fact, in the campus in the Yang, I see a lot of willow trees along the river and so on. This is the type of trees that you find in very damp places. And indeed, the plants use salicylates in a way to protect themselves. And this is really what you have to, as a biologist, you ask yourself, why in the hell plants are making salicylates, are making aspirin and so on. And this was a beautiful experiment published at least 10 years ago in Nature, where they make a tobacco plant genetically modified so that every time the tobacco plant makes aspirin, salicylates, it starts labeling in beta-galactosidase so that they become you. Then they infected the tobacco plant with the tobacco mosaic virus, You see the lytic places here. And what happens is that immediately after the infection, the plant starts making aspirin and brings aspirin where the virus has infected cells. And in these places, aspirin induces apoptosis. So aspirin can induce apoptosis of the sick cells. It gets even better because from the leaves, aspirin is released. The winds spread around aspirin. Other trees in the neighborhood have sensors to feel the presence of aspirin in the air, and they start making their own aspirin. So they are prepared to the upcoming attack. So this was the cover, in fact, of Nature, Airborne Early Warning in Plants. It's a fantastic system how plants indeed defend themselves, not with antibodies like we do, but indeed with salicylates and other molecules like that. And what we think the major working of aspirin is in the anti-inflammatory pathways. Aspirin is a non-steroid anti-inflammatory drug, and what it does, it inhibits these two enzymes, COX-1 and COX-2, cyclooxygenase 1 and cyclooxygenase 2, that are the key enzymes in the formation of prostaglandins, and prostaglandins are obviously very well-known inflammatory molecules and so on. But in fact, if you search the literature with aspirin, it's quite amazing how many things aspirin does. Aspirin, you just mention a random pathway, aspirin will be probably involved in it. We even know that aspirin inhibits the Wnt beta-catenin signal, for example. So that could be a nice, interesting working as a drug in cancer. Even better, we know that aspirin inhibits interleukin-4 production. And interleukin-4 was found some years ago by an Italian group to be the molecule that makes colon cancer stem cells resistant to DNA damage-induced apoptosis. Hey, this starts making sense. So we know that obviously aspirin is a preventive molecule. People taking on a regular basis aspirin are preventive from colon cancer. But now we are thinking, can we also use aspirin as a therapeutic agent to cure cancer, not only to prevent it, but also to cure it. So, and even better, this is an image of circulating tumor cells in breast cancer. And you know, circulating tumor cells are the cells that, through our blood, form metastases. But very often, they are not single cells, they are big clusters of cells. And this cluster of cells are kept together by platelets. Well, if you know what aspirin do, you will realize that indeed, because aspirin is an anticoagulant and so on, it could also be that it will also dissociate this cluster of circulating tumor cells, and in this way prevent metastasis from our patients. So what we are saying is that when chemotherapy is applied to a cancer patient, what happens is that chemotherapy will kill here all the yellow cells, but it will retain the blue cells, which are the cancer stem cells, because they are resistant to it. So in fact, what chemotherapy is doing is a relative enrichment in the proportion of cancer stem cells in your tumor. But now think of combining cytotoxic chemotherapy with, for example, an anti-interleukin-4 therapy and so on, or a simple tablet of aspirin. Remember, one aspirin, I don't know in Chinese valuta, but one aspirin tablet costs 1 cent of a euro, so nothing. It's completely cheap. There's no money to be made with aspirin and so on. That's why Big Pharma hates me, because we are proposing something that possibly can cure cancer in our patients, but without any additional cost. Now, the Netherlands, like Stein knows, is a great country because this thing keeps following me. I will take that. In the Netherlands is a small country with a lot of people, 60 million inhabitants in a very small country, and we register everything. Every time you go into a hospital, everything, we know everything of you, and everything is registered and so on. So we can do a retrospective study now among more than 7,000 colon cancer patients and look at their survival throughout the years, just judging on whether they took aspirin before, after, and during the therapy. And this is our baseline. This is the survival with people not taking any type of aspirin or any other non-steroid anti-inflammatory drug. And look at this. These are the two curves of people taking aspirin after diagnosis or before and after diagnosis. They are significantly doing better than the people not taking any aspirin. This is a retrospective study, so you have to take it with a grain of salt. Interestingly, there was also a group of patients that were doing worse, even though they were taking aspirin, but we found out they were genetically resistant against aspirin. So it doesn't matter, these people cannot be helped by aspirin anyway. But in the majority of the cases, the retrospective study was pointing to a positive effect of aspirin on survival. So regular aspirin use after the diagnosis of colon cancer is associated with a lower risk and longer survival. So can we use aspirin as an adjuvant in colon cancer therapy? When we published these first things, a lot of publicity was made in several medical journals, JAMA here and so on, indeed proposing aspirin as a novel adjuvant therapy for colon cancer. And obviously this was followed by a big trial which is still going on. This is called the aspirin trial, a trial that we are doing in the Netherlands, multiple centers and so on, collaborating with a number of oncologists, surgical people, and indeed, like myself, molecular biologists. And we are simply giving 85 milligrams of aspirin together with chemotherapy. Something that anyway will not harm these people. And in 3 or 4 years, I will be able to tell you whether this is working or not. Thanks God, we are not the only one doing this. There are now aspirin trials all over the world. In China, there is the ASCOT trial and so on. But indeed, as you can see, all over the world, we are testing the working of aspirin as a potent therapeutic agent in cancer. I came to the end of my story. I hope I didn't annoy you too long. I have to acknowledge specifically 2 postdocs, one PhD and a postdoc in my lab, Mark Schmidt and Matthias, that are indeed the first author on the paper that I just sent before coming here. And this was also done in collaboration with the lab of Hans Clevers. Thank you very much for your attention. And as I told you, I'm always advertising for my lab, we have currently 2 positions available, one for a PhD and one for a postdoc. The PhD student will be on oral cancer. The stem cells of your tongue are the most amazing stem cells in the world. I don't know, I think in China you don't do a lot of piercing of your tongue, right? Well, one of the things that we know about the people that have piercing in their tongue is that if you take out the piercing, within 2 hours, the hole is filled. And that gives you an idea of how proliferative the stem cells of the tongue are. And these are the stem cells that also cause oral cancer. So we are very fascinated by this oral cancer, and this PhD student will work at that. And the postdoc, as I told you, will work as Paneth cells as a cell of origin of cancer in the future. So if you're interested, you just have to visit my website where this position will be advertised. Thank you very much for your attention.
Well, thank you for that very interesting lecture. I'm sure there are many questions. So let's first give the opportunity to the students before we ask
if they dare.
the staff to start asking questions. Any questions for Professor Fodde?
Hi, Professor. A very wonderful talk. Thank you.
Thank you.
And I was wondering whether someone has published some paper that the Paneth cell can act like a cell of origin, at least to the organoid. And another small question.
No, it's okay.
Sorry.
Go ahead.
And I noticed that when you introduce your DSS model, you also elevate the LGA5, elevate the ability of LGA5 positive cells to generate organoid. How does that happen?
Now, okay, I will start with the second part of your question. It's the opposite. Upon DSS, LGA5 are eliminated.
Sorry,
that
PowerPoint, that certain PowerPoint.
Yeah, I'll show you the corresponding bit of the PowerPoint. If I can find. Yeah, this is it. You see here, the stem cells are in green here. This is the control, and that's the DSS. So they are gone from here to here. So there is a dramatic ablation of Paneth cells. And this is because, indeed, and it's compensated by Paneth cells that become stem cells and so on.
I got it, but
Okay.
the later one PowerPoint, if you would, yeah,
You mean following up this?
following on this.
This part?
No, not
Oh.
this one.
You have to tell me where.
No, not even this one. It's like a few bars. Not this one, maybe.
No, this is about SCF, so it must be either here or here. But tell me what you want to say.
In your DSS model, I noticed that one computation diagram, I noticed that the DSS actually elevate the ability of LGR5 positive to generate odd noise.
No, it's.
So that's the slide.
Oh, I think you are referring to this one. But here we're using Paneth cells from inflamed mice, and we associate them with stem cells from normal mice. So the LGR5 stem cells there are coming from a normal mouse. When I put them together with the Paneth cells from the inflamed mouse, they do better. Yeah?
Sorry, I still don't get it. Basic PowerPoint, its name is How Do Paneth Cells React to Information?
That's it?
Yeah, this one, and the upper middle.
Yep.
Yeah, there is actually LGR5 DSS. Yeah, LGR5 DSS, and that is actually higher than the LGR5.
So LGR5 DSS plus Paneth cells DSS.
The last
is
but
high.
one. The last but one. Yeah.
This one.
The last but one. Yeah.
This one.
Yeah.
Yeah, but that's nothing. That's really 2 or 3 organelles.
So.
So they are not doing better than, oh, you're comparing this one with this one?
Yeah.
Yeah, it's like, 1 or 2 here, 2 or 3 here. They are not significantly different. Statistically, it's not a difference. You have very sharp eyes, but indeed, look at this. I mean, I hope you notice also this part. This goes really way up. So that is the important, significant part. That LGR5 still.
So you should compare this one with this one. It goes much further.
Yeah.
Compared with this one and this one, not much different.
The thing is that when we do lineage tracing, you see there are very few LGR5 left over. The ones that are left over can work as stem cells. So qualitatively, they are good. They still can do the job, but they're just gone. You see, these are LGR5 in control, and these are LGR5 in inflated mice. So they are not there anymore. They cannot do the job. The few ones that are still there can still do something. And that's how you explain those organs that you very sharply see. But in general, it's really, there must be something else that compensates this huge loss of stem cells. You also had the first part of the question. I hope not. So you were asking whether someone else is publishing Paneth cells as a stem cell and cell of origin of cancer. Well, I hope not, because most of the research in my lab will be. So it will be a race as usual. Other people in the world will compete with us to do this, but I hope we will win the race. What can I say? Yeah. Hey.
Long time no see.
Hi there.
Yeah, I wanted to know a question related to this topic and the aspirin. Like in this model, you use DSS and later the inflammation environment. But if you use aspirin, can you rescue the ability of LGR5 cells?
Wait, so you're talking about aspirin?
Yeah, to rescue the ability of LGR5.
Oh.
Renew cells.
Actually, I don't know about that. So aspirin we are using in the context of cancer, not only in the context of
Yeah,
inflammation.
because the DSS makes the inflammation environment.
Right.
But if you use the aspirin
Some
to
gastroenterologists, I don't know if you can confirm this, you are a medical doctor yourself.
Yeah.
Some gastroenterologists told me that in general you don't give aspirin to Crohn's disease patients.
Yes,
or,
yeah.
So it's not done. For some reason it's not good for them.
Yeah.
But I'm not talking about that. I'm talking about indeed aspirin to cure cancer and so on. not to cure inflammation.
Yeah,
I know that aspirin is an
aspirin
anti-inflammatory drug.
just a drug, maybe another drug to remove the inflammation
Yeah.
environment.
I think that's a good point. It can be tested in the mouse and so on. So I will thank you for the suggestion.
Any other questions?
Yeah, here.
Fantastic.
Thank you
So
very
my
much.
question is, why the nature evolve or need such kind of special mechanism?
Ah, you mean like the lizards and the
Yeah,
chop
when
up
the
the
energy
yeah.
plus going down, and when the stem cell to the stem cell.
I have a very simple answer to that. Evolution does not work on our capacity of regenerate a limb or an arm or a leg. Evolution works on our reproductive fitness. What drives evolution is our capacity to generate children, right? And that has nothing to do with the capacity of regenerating an arm when it's chopped up. So I don't think that the regenerative capacity is an important issue in evolution. I ask myself the same question. Obviously, it's my opinion against any other opinion. There is no answer to that and so on. You can still speculate, but I love the question. Fish have that, amphibians have that, frogs, salamanders, lizards and so on, but the mammals don't have it. So fascinating. Yeah.
Nice talk. So my question is, the inflammation will kill the LG5 positive intestinal stem cell,
Yeah.
but the parent cell will dedifferentiation into a stem cell-like cell.
Right. Correct.
Are they similar, or these kind of two stem cell-like cell is the same?
So in the long run, there
What
will.
difference or what's the same between the two?
In the beginning, they are different.
Yes.
But after, if we wait like 5 to 7 days, we see that they acquire LGR5 expression. So they become LGR5 cells. And everything is back to normal.
Okay, they will put. They
And
also
then
will
in the long
express
run, not immediately, but in the long
In the
run,
long run also the
yeah,
same.
yeah.
Did you do some, genome sequencing or proteomic, tracking for the two different population of cells after?
That's a
inflammation?
very good one. No, we didn't do, but that's a very nice idea. It will be an
Even
expensive
single
experiment.
size
but, yeah.
sequencing.
Yeah.
But maybe this.
Probably I wouldn't do sequencing, but more RNA-seq, so expression,
I see,
gene expression.
yes. But I think maybe they're not. It's heterogeneous. I think.
But I don't think
Not
it's
homogeneous.
I agree with you, but at the gene expression level, not at the genomic level. I don't think that there is time to acquire mutation and so on. I had a very interesting lunch discussion with Benji about the relative importance of genes versus the environment and so on. You know, I used to be a geneticist. I used to believe very much in genes and how important genes are. Now I think that epigenetics is much more important than genetics.
Thanks.
Any other questions?
So I have a question about the past cell rotation. As you know, the stem cell, LGR5 stem cell can differentiate to the past cell and it will go up, and how it go down to differentiate to the past cell and located between the stem cell, LGR5 positive stem cell. Do you have any
I'm not sure I understood the question. You say Paneth cells are derived from LGR5 cells, right?
Yeah.
Yeah?
So it's differentiated to the LGR5, why it's well located between two LGR5 stem cells. It's usually like the epithelial cell and go up to the crypt.
So, if I understood well your questions, I think that the reason are different levels of Wnt signaling. So we know that when Paneth cells differentiate from LGR5 cells through Wnt signaling, which is very weird because I told you that Wnt signaling is a signaling of stemness, but there is one example where Wnt signaling can cause differentiation, and that is indeed from LGR5 to Paneth cells. Paneth cells have a very nice nuclear beta-catenin pattern, unique. But it's one level of Wnt signaling activation. If I activate Wnt signaling even more, then the Paneth cells become an LGR5 cell. So there are usually in textbook, your students read about signaling the pathway, and it's like an on or off. Either they are activated or they are not activated. I think dosage is also very important. For each pathway you have lots of dosages, and for each dosage there will be a different outcome in the cell and so on. So it's very subtle, very different in dosages. I hope that that was your question.
What I thought is how the stem cell is move like the TA cell go up, and how the stem cell move to like the PAT cell located on between the two stem cells, not go up like the TA cell. I'm not sure why you got that.
No, probably it's just a language problem that I cannot follow you entirely, but I'm guessing.
Not the winter signal. I'm interested in the location of the PAT cell.
Yeah, but that's why location is important in wind. Location is what I was. Yeah. Imagine that here there is the source of wind signals, right?
Okay.
So there is a gradient. Wind signal will diffuse through the tissue and will form a gradient. These cells here will be very close to the source of wind, right? But transient amplifying cells will be a little bit more distant. So this will be all different levels of wind signaling activation.
So I changed my question to like this. The stem cell will differentiate to the TA cell.
Yeah.
Yeah. So the stem cell also is directly differentiated to the PAT cell, or it should like to go to the TA cell then to the PAT cell.
So the question is whether first an LGA5 cells form a transient amplified cells and then a PANET cell.
Yeah. So it's how to move to the bottom of the crystal.
Okay, there are not definitive evidence on this. People think there are some partial evidence that PANET cells are directly derived from
Stem cell.
stem cells,
Yeah.
not through a transient amplified cells. We made it.
Yeah.
I finally answered your question.
So if there are any
Sorry
published-
about it, it's my fault.
If there are any published, I have not found any published that the LGR5 stem cell directly.
Yeah, there are partial evidence for that, but not complete. So it's a very good question.
Yeah.
Yeah. Thank you very much.
Thank you.
Any other questions? I have one. So I'm a microbiologist, and of
Oh
course
my
you talk
God.
about the gods, how full of microbes.
Nobody's perfect this time.
No, I know. But so it turns out more and more that microbial, disbalance in the gut causes many of the problems,
Yeah.
including, IBD, etc.
Yeah.
Of course, many microbes, they activate the ATP pathway as well. So how do you think microbial impact on this? And can microbes be used actually to cure the cancers by, getting rid of all the microbes first and then add the microbes that specifically do stuff? Yeah?
So I am starting to do this, and I am understanding how complex this piece of science is, and so on. To study the microbiome is hell because of the complexity. So we know that inflammatory bowel disease have a different microbiome. We know, for example, that lactobacillus is going down; it's not present anymore, both in inflammatory patients and also in patients eating Western-style diet and so on. So we know that there is a difference in microbiome, and we exactly know which strains are absent or present in the two groups. My problem as an experimental scientist is, yes, but what do I do with it? How can I prove? Because it's such a complex experiment. Lactobacillus is one of millions of different strains that are populating our gut. I can make, obviously, mice sterile and infect them only with lactobacillus, but what kind of experiment is that? It's only lactobacillus without the rest of the microbiome and so on. So what we are doing is using the metabolites that are made by specific strains. For example, we are adding lactate into organoids, and we see indeed a huge response to lactate and so on. So the stemness is increased by using lactate and so on. So yes, my hope is, and I don't know if I can contribute to this because of the complexity of the scientific problem, but yes, I don't know whether people will use bacterial strains or metabolites to cure their inflammation and or cancer and so on, but I certainly believe that that will be the way, and that the microbiome is playing an essential role as a niche of the stem cell in the gut. Very difficult to study.
Yeah.
Very complex. Okay.
Any other questions? Well,
Thank you very much. Thank you.